Imagine the inside of your cells, not as this perfectly ordered static factory, but as a frantic, chaotic metropolis.
Right. It's incredibly noisy in there.
Exactly. Right now, as you're listening to this, millions of microscopic proteins are sprinting through your DNA.
Yeah, these transcription factors and chromatin remodelers, they are constantly flying around.
Frantically docking onto your genetic code just to turn specific genes on or off, and then, boom, popping right back off a fraction of a second later.
And honestly, it is a staggering amount of traffic. Capturing that dynamic process has been, well, one of the greatest technical hurdles in modern biology.
Because these interactions are incredibly brief. Very brief. Okay. And physically, they are very weak. Yeah. I mean, we are talking about transient connections that disappear almost as soon as they form.
Which brings us to the mission of our deep dive today. We are unpacking a groundbreaking 2026 paper from the journal Cell titled Single-Cell Mapping of Regulatory DNA-Protein Interactions.
Yeah, the researchers here have solved a massive, massive problem with how we study this microscopic metropolis. Because if you look at the current mapping methods, things like ChIP-seq or CT and tag, they act like cameras with incredibly slow shutters.
That's a great way to put it.
Like they are trying to photograph molecular paparazzi that are just sprinting past the lens. To get even a blurry picture, these old methods require millions of cells.
Millions. Or even worse, they have to use these incredibly harsh, salty chemical washes just to prepare the samples.
Which just ruins the very thing you're trying to look at.
Exactly. Yeah. The high salt conditions are notoriously problematic for biologists. I mean, the intent behind the salt wash is to strip away the background noise of random floating proteins.
But the collateral damage is immense.
Right. The salt literally breaks the delicate physical bonds between the transcription factors and the DNA.
Wow. So we're just destroying the evidence.
Pretty much. So for years, we have only been able to map the strongest, most stable proteins.
While the elusive ones, the proteins that often act as, the critical master switches for disease or human development, they remain completely invisible to us because our own tools just wash them away.
But the solution presented in this paper completely flips the script. The researchers developed a new method called D&D-seq.
And rather than trying to take a slow shutter photograph, D&D-seq acts like a molecular stamp.
Yeah, it leaves a permanent physical footprint directly on the DNA molecule itself,
marking exactly where that fleeting protein was sitting inside a single individual cell. Okay, let's unpack this. How does a molecular stamp actually work on a chemical level?
Well, the elegance of the D&D-seq mechanism is really in its modularity. D&D-seq stands for docking and deamination, followed by sequencing.
Docking and deamination.
Right. To understand the stamp, we really have to look at the ink. The researchers utilized a base editing enzyme called DDDA11.
And a base editor is basically a molecular machine that can chemically alter the actual letters of your DNA, right?
Exactly. They fused this DDDA11 enzyme to a secondary nanobody.
Can you explain what a nanobody is for us?
Sure. You can think of a nanobody as a tiny, highly specific homing beacon. It's programmed to target an antibody that is already sitting on your transcription factor of interest.
Okay, let me visualize this. The transcription factor naturally docks onto the DNA. Then an antibody grabs hold of that transcription factor.
Yep, you got it.
And finally, our engineered nanobody homing beacon arrives, carrying this base editing enzyme right to that exact spot on the genome.
Precisely. And once it is perfectly positioned, the DDA11 enzyme performs the deamination step.
What does that mean chemically?
It basically chemically removes an amino group from a specific DNA-based cytosine, or C, which converts it into uracil, or U.
Wait, but uracil doesn't normally belong in DNA, right? I thought that was the building block of RNA.
It is. That's the trick. So when scientists later run that DNA through a sequencing machine, the machine reads that anomalous uracil as thymine, a T.
What?
Or if the sequencer is reading the opposite strand, a G appears as an A. So a C to T or GA edit becomes a permanent readable stamp.
Proving, without a doubt, that the protein was sitting right there.
Exactly.
So I'm thinking of it like a molecular stapler, like it docks into place and it deaminates the base, leaving a permanent staple right in the DNA strand.
That's a perfect analogy. The D&D equals docking and deaminating.
But wait, our cells naturally mutate all the time, right?
They do, yeah.
So if we are just unleashing a base editing stapler into the nucleus, wouldn't it just float around and create absolute chaos, randomly mutating genes everywhere it bumps into them?
That is the big question. How do we know which mutations are from the D&D-seq stamp and which are just, background noise?
Right.
Well, the researchers anticipated that exact problem, and their solution is honestly the defining genius of the entire design.
What did they do?
They didn't just put a fully active enzyme into the cell, they physically split the DDDA enzyme into two halves.
Like they cut it?
Yeah. By severing the protein chain exactly at position 1397, they created an N-terminal half and a C-terminal half.
So they basically broke the stapler in half before sending it in.
Exactly. And because it's split, it is entirely inert.
It can't do anything.
Right. The N-terminal half the part carrying the homing beacon can float around the entire genome, bump into thousands of genes, and it won't mutate a single base.
Because it physically cannot perform the chemistry without the other half.
Yes. It only regains its catalytic power when the researchers intentionally flood the cell with the missing C-terminal peptide, along with a zinc ion cofactor.
Ah, so the zinc acts as like a final locking mechanism.
You got it. It folds the two halves back into a functional enzyme. This split design gives researchers total switch-like control over when the editing happens.
Which completely prevents that chaotic background noise.
Precisely.
And beyond the chemistry, the data itself leaves a very specific signature, right? Because the enzyme is tethered to the side of the protein, it can't stamp the DNA directly underneath the protein center.
Exactly, has to reach around it. So when the researchers sequenced the DNA, they found a distinct bimodal distribution footprint.
Bimodal meaning two peaks.
Right, because the enzyme reaches around the flanks of the transcription factor that edits cluster perfectly in a 200 base pair window.
Just on the edges.
Yeah, creating two distinct peaks exactly 50 base pairs away from the center of the binding site on either side. It forms a perfect symmetrical footprint.
So let me just do a quick bulleted recap for you before we move on.
Sounds good.
First, standard methods completely fail at catching these weak DNA interactions because they wash them away.
Correct.
Second, D&D-seq uses a brilliantly engineered split enzyme to edit C to U right where the protein sits.
Yes, the broken stapler.
And third, this leaves a permanent symmetrical footprint on the DNA that we can easily read.
A perfect summary.
So here's a question for you to mull over. If you had a molecular stamp to track any invisible event in your own body, what would you track first?
That's a fascinating thought.
It is. But moving from bulk populations of cells to resolving these invisible events inside single cells, that is where this technology truly forces a paradigm shift.
Oh, absolutely. To prove D&D-seq could work in single cells without creating a leaky, cross-contaminated mess, the researchers designed a fascinating sorting experiment.
The sorting hat experiment.
Right, like from Harry Potter, they took two completely different human cell lineages.
They had CA46 cells, which are lymphoid immune cells, and K562 cells, which are erythroid cells, like early red blood cells.
And they mixed them equally in the exact same tube.
But before they mixed them, they did something clever.
Right. Yeah, they stained the CA46 immune cells for a protein called CTCF, and they stained the red blood cells for a master regulator protein called GATA1.
Okay.
Then they ran the D&D-seq reaction on the whole mixed population.
So the logic is simple. If the split enzyme method was leaky, if the staplers were reassembling and floating around stamping random DNA, you would see a mess.
Exactly. You'd see GATA1 footprints inside the immune cells or CTCF footprints inside the red blood cells.
But the data separated perfectly into two distinct clusters. Zero cross-contamination.
None at all. And not only did it separate perfectly, but it completely outperformed the existing gold standard methods in terms of signal clarity.
Let's talk about those benchmarks.
Sure. When evaluating genomic technologies, one of the primary metrics is the FAFR-P score, which stands for fraction of reads and peaks.
And that's basically a direct measure of the signal-to-noise ratio, right?
Exactly. A high FAFR-P score means the vast majority of your sequencing reads are landing exactly in the actual binding peaks. A low score means your data is mostly random background noise.
And the numbers from the paper are wild.
They really are. D&D-seq achieved a FAFR-P score of around 60%.
60%.
And it held that incredible 60% clarity across 5,000 cells, down to 500 cells, down to 50 cells, all the way down to a sample of just 10 single cells.
While the competing state-of-the-art method, which is called Yilicki-Tiner UN, hovered around a 14% FAFR-P score.
Yeah, meaning 86% of the competing method's data was essentially garbage noise.
That is a massive upgrade.
It gets better. To widen that gap even further, the researchers built a sophisticated computational algorithm to filter their data.
A logistic regression model, right?
Yes. They trained this model on the bulk D&D-seq data to learn the mathematical properties of a true on-target footprint.
And a logistic regression model basically acts as a mathematical gatekeeper. It evaluates all the variables and calculates a probability of whether an event is real biology or just a sequencing error.
Exactly. And the algorithm determined that the single most predictive feature of a true binding event was the maximum number of edits in a 100 base pair sliding window.
A sliding window, so physically scanning along the DNA strand.
Right. Because D&D-seq physically tethers the enzyme to the protein, the deamination edits are intensely clustered. Natural, random cellular mutations are scattered thinly across the genome.
Oh, I see.
By sliding that 100 base pair window across the DNA sequence and hunting for the highest density of edits, the algorithm perfectly separates biological signal from random noise.
Here's where it gets really interesting. What was the accuracy score on that?
It achieved an area under the curve, or AUC, of 0.84.
And just for context, in statistics, an AUC of 0.5 is basically a coin flip, right?
Right, and 1.0 is perfect. So 0.84 indicates a highly accurate, robust predictive model.
Okay, let's do another quick bulleted recap.
Go for it.
Number 1, D&D-seq works flawlessly in single cells without cross-contamination.
Verified by the sorting hat experiment.
Number 2, it beats older methods with massive 60% RAP scores. And number 3, an algorithm filters the data by looking for dense edit clusters in 100 BP windows.
Spot on.
So here's your next question. How might an algorithm that so perfectly separates signal from noise change the way we diagnose diseases?
That is exactly where the researchers went next. Knowing we can filter out the noise in healthy cells is a great proof of concept, but the true test is whether it can catch a disease before the cellular architecture completely spirals.
And that brings us to the CHIP patient data.
Yes. The researchers applied D&D-seq to a real patient sample with a condition called clonal hematopoiesis of indeterminate potential.
It's a mouthful. But CHP essentially means a patient has a mutant clone of blood cells that are beginning to take over.
Right. While it isn't full-blown cancer yet, it is a massive risk factor for leukemia.
And this specific patient carried a very specific mutation called IDH2-R140Q. Can you walk us through the mechanics of that?
Absolutely. To understand why this specific mutation is so dangerous, we have to trace the metabolic cascade it triggers.
Okay.
When the IDH2 gene mutates, the cell begins to synthesize massive, unnatural amounts of a metabolite called 2-hydroxyglutarate.
So it's like a microscopic factory that suddenly starts overproducing a toxic byproduct, flooding the factory floor.
Exactly. This 2-hydroxyglutarate builds up and chemically blocks a family of enzymes known as TAE enzymes.
And what do TAE enzymes normally do?
They are the genome's molecular erasers. Their entire job is to remove bulky chemical tags called methyl groups from the DNA.
Oh, I see. So because the TAE enzymes are incapacitated by the toxic byproduct, the DNA becomes hypermethylated.
Right. It becomes densely covered in these physical chemical tags.
And those methyl tags act as physical roadblocks on the DNA strand.
Yes, and the crucial protein that gets blocked from docking by all these roadblocks is our old friend, CTCF.
To capture this entire catastrophic sequence happening simultaneously in a single cell, the researchers utilized a dual method approach, right?
They did. They combined D&D-seq with a technique called GATI-CHA, which let them simultaneously sequence the targeted mutations and measure the physical accessibility of the chromatin.
All in the exact same CD8 T cell.
Exactly, and they proved definitively that the mutant T cells had severely reduced CTCF binding compared to healthy cells extracted from the exact same patient.
It is hard to overstate how devastating losing CTCF is to a cell.
It really is.
Think of your DNA as a massive skyscraper. The CTCF proteins are the internal load-bearing walls.
A great analogy.
They create these isolated rooms and hallways in the genome, which biologists call topologically associating domains, or KDs.
And these walls provide insulation. They ensure that an activation switch meant to turn on a harmless gene in one room doesn't accidentally reach across the hall and turn on a cancer-causing gene in the next room.
But by losing CTCF, you are systematically knocking down those load-bearing walls.
Precisely. Using advanced predictive algorithms called C-origami and Cicero, the researchers modeled the 3D consequence of this loss.
And what did they see?
They demonstrated that without CTCF, the three-dimensional chromatin structures completely collapsed at specific genetic loci.
Like the GIT1 and CNIH2 genes, right?
Yes. At those genes, the highly organized, distinct leaps of DNA degraded into a disorganized tangle.
The walls fell down, and the rooms merged into chaos.
It is actually worse than just merging. New rogue networks formed.
Rogue network!
Yeah, without the insulation provided by CTCF, Distant pieces of DNA that should never touch began communicating.
Oh, wow.
They documented this happening with the UFIP2 gene, where aberrant loops formed, completely rewiring the cell's genetic architecture.
It is terrifying to think about, really.
It is.
So time for another quick bullet recap. Number 1, a specific IDH2 mutation blocks the CTCF protein from binding.
Right, because of those methyl roadblocks.
Number 2, D&D-seq captured this loss in single mutant cells. And number 3, without CTCF, the 3D structure of the DNA collapses, rewiring the cell's genetic architecture.
Perfectly summarized.
So here's a question for you. Can you imagine how a single missing molecular wall could completely change the function of an entire cellular room?
It really shows how fragile the whole system can be.
Yeah. And we don't just want to look at the static aftermath of these collapses,
Yeah.
right? We want to watch the biology unfold over time.
Which leads to the final and perhaps most dynamic experiment in the paper. The researchers conducted a 24-day time-lapse experiment mapping erythropoiesis.
Which is the birth and maturation of red blood cells.
Exactly. They took CD34 plus hematopoietic stem cells, basically the raw, undifferentiated clay of the blood system.
And biochemically coax them into maturing into red blood cells over the course of 24 days.
Right. And throughout this 24-day journey, they mapped the footprints of GATA1, which acts as the master conductor of this whole process.
Tracking exactly where it stamped the genome.
Yes, while simultaneously tracking the RNA output of its downstream target genes, such as SLC4A1 and RHA.
Which are genes that are critical for the cell to actually function as a red blood cell.
Precisely.
And this leads to an incredible aha moment. Because when they aligned the timeline of the footprints with the timeline of the RNA output, they found something completely unexpected.
Something that challenges the standard textbook model of gene activation, yeah.
What do they find?
They discovered that GATA1 binding and the downstream gene expression are temporally decoupled.
Meaning they don't happen at the same time.
Right. We have traditionally assumed that as soon as a transcription factor docks onto a gene's promoter, the gene immediately turns on and begins transcribing RNA.
Like flipping a light switch.
Exactly. But the D&D-seq footprints prove that GATA1 docks to the DNA long before the actual gene activates.
Wait, really?
Yeah. The protein arrives, binds to the regulatory element, leaves its permanent CDU footprint, and then just sits there.
For how long?
Sometimes for days. The actual gene expression follows much later in the differentiation process.
So what does this all mean?
It means the protein is basically setting the stage. It arrives, locks in, and waits.
And if we were only using traditional RNA sequencing to study this process, we would completely miss the true beginning of the story.
Entirely. We would look at the RNA output on day 15, see the gene turning on, and we'd be completely blind to the fact that the GATA1 protein actually arrived and prepared the site on day 5.
Because RNA sequencing only gives you the final output.
Right. D&D-seq gives you the underlying regulatory blueprint. This poised state likely provides an immense evolutionary advantage.
How so?
Well, by preloading the transcription factors days in advance, the cell holds the genetic locus open and ready.
Like a bookmark holding the page open.
Exactly. It allows the cell to respond instantaneously to a final developmental cue or an external stressor without having to assemble the entire complex from scratch.
Which means older technologies have essentially been looking at the final act of a play and assuming the actors just magically appeared on stage.
Well, D&D-seq lets us see them waiting in the wings for days.
Incredible. Okay, let's do our final recap. Number 1, blood cell differentiation was mapped over 24 days.
With GATA1 as the master switch.
Number 2, D&D-seq proved that proteins bind to DNA days before the genes actually activate.
Yes, the temporal decoupling.
And so here's a question for you. If a protein binds to your DNA, but the gene doesn't activate until days later, what do you think that protein is waiting for?
Synthesizing everything we've discussed today, this technology truly is a paradigm shift in how we interrogate the genome.
It really is.
For decades, our understanding of molecular biology has been heavily constrained by our tools. We were taking slow, blurry snapshots of populations of cells.
Entirely missing the weak, rapid, and poised interactions that govern so much of our biology.
Exactly. By utilizing a split base editor to permanently stamp these transient interactions directly into the genome, we aren't taking snapshots anymore.
No, we're reading the hidden, dynamic diary of the genome.
We can finally see the ghost-like interactions that dictate cell fate, disease progression, and the 3D architecture of our DNA.
It completely changes how we see that bustling metropolis inside our cells.
It really does.
So I want to leave you with a final thought to mull over. If we now have the technology to see fleeting, invisible molecular interactions leaving permanent footprints on our DNA, what other ghost-like events in our bodies are leaving traces we simply haven't learned to read yet?
It's a profound question.
Do you think the emotional trauma of our ancestors, or even a passing viral infection, leaves a transient stamp in the muddy waters of our epigenome, just waiting for a tool like D&D-seq to finally uncover it? Keep exploring, keep questioning, and we'll see you in the next deep dive.